chapter 18 here. All right, so this is blood. Anything and everything you wanted to know about blood? Hopefully, maybe some of it. All right, if you recall back in chapter 5, when you were in Bio 210, we talked about the different types of connective tissue. And all right, blood is one of the last types of fluid connective tissue. There's 3 types of connective tissue proper, supporting connective tissue, and then fluid connective tissue. Blood and lymph are part of that category. So the nice thing about this type of connective tissue is that it is constantly being made. Right? For example, a red blood cell's lifespan is only 120 days. Right? That's 4 months. All right, so it's constantly being renewed. Same with white blood cells, same with platelets, and same with some of the other elements in the blood, because we're not just talking about the cells, there's also going to be some of the other material in there. There's only one diagram to label for chapter 18 in the lymphatic section in the atlas. Yep, that is correct. So again, like I said, there's not a lot of identification material for chapter 18 on there. So the purpose of blood, because it is in the circulatory system, is to move stuff around your body. So it is going to move different things. Obviously, respiratory gases, carbon dioxide, all right, oxygen, tons of different nutrients. Glucose is a great example, lipids, all right, anything that cells can use to help generate ATP. At the same time, you know, the cells are going to be needing it for energy processes and metabolism, but unfortunately, they're going to generate wastes. So we got to get rid of those wastes. And you just got done learning about chapter 17 and the whole function of that chapter, which deals with chemical messengers, i.e. hormones. All right, so all of those are going to be traveling in the blood. So the cardiovascular system plays a big role in that. Obviously, all right, we have our pump, which is the heart, which is going to move this fluid through the circulatory system. And we'll go into it more when we get into, all right, the circulatory system. But obviously, arteries, and you should know this by now, are going to move blood away from the heart. Veins are going to return it to the heart. All right, so, but don't always, you're going to learn when we get into the heart, and we're going to be talking about this more, about the different blood vessels that connect to the heart, right? That not all arteries carry what we call deoxygenated blood, or carry oxygenated blood, and not all veins carry deoxygenated blood. More on that later on. Then finally, capillaries. That's where we mix things. Okay, it's the big mixing pot or bowl or section or whatever. This is where we're going to exchange that blood with the tissues. All right, so these are very small vessels there. All right, so let's look at blood. And so when we look at blood, we're going to break it down into 2 parts. One, what we call the formed blood elements, you know, the actual physical structures. And then there's the plasma, the liquidy part. Okay, so our formed elements include our red blood cells, the white blood cells, and the platelets. That will be erythrocytes, leukocytes, and platelets. We'll talk about some of their functions in this course here. All right, the red blood cells are going to transport those respiratory gases, and we get to talk about, all right, the exchange of respiratory gases more so when we talk about the lungs. All right, how oxygen and carbon dioxide are going to move in and out of the blood and in and out of the tissues. And then obviously leukocytes, we hear a lot about those, you know, when we talk about the immune system. All right, and so these white blood cells are going to help us, all right, in our immune response to certain pathogens. And then of course, if you do compromise a blood vessel, all right, well, we need to repair it, stop the bleeding, and so our platelets help to prevent blood loss by kind of clogging things up. And then, of course, we have our plasma, which is the fluid portion of our blood. And this is where we're going to see lots of plasma proteins, albumin and the sort. Lots of proteins that are carriers. They might bind with a hormone, all right, or a certain substance and move it from point A to point B. We learned back in chapter 10, calmodulin is a wonderful calcium binding protein, and calsequestrin, all right? And so these are just examples of calcium binding proteins. But again, we'll see other types of proteins in our blood that will bind onto certain substances. Also, this is where stuff goes to dissolve, All right, a lot of our substances will dissolve right into the blood. It's something that's water soluble, dissolves right into the blood, gets transported around. So talking about transportation, all right, here's what we're going to see with our functions of the blood. Okay, this is important. Obviously, I mentioned this before. Our blood is going to move things around your body. All right, we talked about how it's used in the respiratory gases for carbon dioxide and oxygen. We already mentioned, all right, how it carries nutrients out to the tissues and takes waste from those tissues, how we transport hormones from the gland that actually synthesizes the hormone, then secretes it, and then it carries it out to the target cell. So we see all that. And you're going to find an interesting fact about heat here with blood is that your blood temperature is usually about 1 degree warmer than your regular body temperature. So it's important because if you're too hot, then we take the blood, undergo vasodilation in a lot of the blood vessels in our skin, and it moves the blood towards the surface of your skin so that heat that it carries can be given off. And vice versa, in cold temperatures, we shunt the blood away from the surface of the skin because we want to retain that heat. And so we'll see vasoconstriction. Protection, that plays a big role with our immune function. All right, so the leukocytes will be a part of that protection. The plasma proteins, same thing. Those proteins, when they bind on to certain components, like for example, hormones, and a lot of substances like drugs, for example, you may have heard the term half-life before. That's basically how long a drug is going to usually last for as it starts to degrade. Well, plasma proteins will help with certain hormones. When they bind onto the certain hormones, it helps to extend their half-life so they don't degrade as fast. So that's also important when we're talking about the functions of blood. All right, and then of course our platelets help against blood loss, and we'll also see again, those plasma proteins will also help prevent against blood loss also. So when we're talking about functions of the blood, regulation of several different body conditions, you heard me talking about body temperature just a moment ago, all right? That is an important component there, all right? As tissues are undergoing metabolism, I don't want to bore you with the different types of chemical reactions, but a lot of our chemical reactions in our body are what we call exothermic. And as a result of these chemical reactions, they give off heat. And so that heat has to go somewhere. Exercising muscles all the time. As you're exercising, they start to warm up. All right. So the blood will help to take that heat away from those body cells and distribute it, all right, to other areas depending on what's happening, if you're too hot or if you're too cold. All right. But like I said, a great way to get rid of that heat is through the blood vessels that reside in your skin. I mean, for God's sake, the skin is the largest organ in the body, so let's utilize it. Body pH, huge, huge role here. All right, back in chapter 2, you learned about acids and bases. All right, substances that will release hydrogen ions or absorb hydrogen ions, the H+. All right, so this goes on with certain metabolism processes in the body. And so in our blood, and you will learn about this later on in the course, we're going to talk about chemical buffers. Remember, a chemical buffer is something that maintains the physiological optimal pH. And if you don't remember what that range is, all right, our range for your blood pH is going to be 7.35 to 7.45. So ideally, what we're looking for is going to be 7.4. And if you recall, that on the pH scale is roughly about, it's slightly acidic, basic, more alkaline. So you have these buffers that reside in the blood. And so when your blood starts to become too acidic, these buffers will try to prevent, all right, the pH from getting to dangerous acidic levels. Doesn't want the pH to drop too low. We don't want it to fall below 7.35. Because when that happens, when your chemical environment becomes too acidic, you can start to undergo what's known as protein denaturation. They just start to break down. They start to unfold. And then basically that compromises the functions of proteins. You compromise the functions of proteins, you're going to have some issues going on there. All right, so keep that in mind. So it helps and plays a huge role, all right, in our in our chemical body pH. Fluid balance, again, we'll talk more about this later on, but as you know, all right, water concentration in our body is crucial. You've seen it yourself when you're going to the bathroom, all right, when you're peeing. If you notice, right, if you haven't drank for a while and it's really hot and you become dehydrated, your urine, it looks more like a iced tea color, right? That means you're dehydrated. And so, you know, we have to keep in mind that our water balance is crucial because you use water for a lot of things in your body, all right? So as you're drinking water, water will go from the GI tract into the blood. And then also, as you're exercising, as you're doing certain activities, you'll lose water through sweating, urination, all right, through exhaling. Okay, you breathe off some water vapor. When you are actually defecating through some of the fecal matter, there is a certain amount of water there. So again, we're going to lose water and gain water, all right? But it's important that we understand that a lot of this exchange of water that occurs between our blood and the other tissues of our body, like our GI tract, respiratory tract, all that, okay? That's what we're meaning when we talk about the interstitial fluid. We need to maintain what we call this osmotic balance. Because if you recall, water goes from an area of high water concentration to an area of low water concentration. It's a follower. It likes to follow solutes. Glucose is a solute. Sodium is a solute. So that can influence, all right, where water travels. So certain proteins that are going to be found in our blood, and we'll talk about it, like albumin, and certain ions, I just mentioned one, sodium, is going to help influence the movement of water in and out of the blood. All right, so let's take a peek at blood. So we're looking at blood, and you've probably seen it when you've given blood or they've had to lab test your blood, okay? The thing that jumps out is usually the color. And so when we're talking about the color, that depends on the degree of what we call oxygenation, how much oxygen is in that blood. So if you see that your blood is bright red, then we assume that it's oxygen rich. Arteries will have oxygen rich blood, okay, for the most part. Oxygen poor blood will appear to be a dark red, right? So we use terms like oxygenated and deoxygenated blood. Just because I'm using the term deoxygenated blood doesn't mean that it does not have oxygen, it just has very low levels of oxygen. Your veins do carry oxygenated blood. Excuse me. Your veins carry, all right, blood that does have some oxygen in it. It's just low levels, okay? So it's oxygen poor blood. So when we talk about how much blood is in the body, we're going to use volume to describe that. And most adults, all right, have about 5 liters, okay? Men slightly more because men are slightly larger than women. All right, viscosity, cool term. Talks about how thick the blood is. Okay, you can see here, blood is 4 to 5 times thicker than water. All right, and it really depends on how much stuff is in the blood. When we talk about our viscosity, all right, what we're talking about is, all right, the formed elements that are in the blood and the amount of dissolved or suspended substances that are in the blood plasma. Okay. So if you were severely dehydrated, the water content of your blood plasma is most likely going to be pretty low, which means, okay, that the ratio between the blood elements, formed elements, and the plasma is going to be in favor of the blood elements. So your blood will be thicker. So that means the viscosity increases. And we'll see that, for example, with our red blood cells. All right, one of the dangers of climbing Mount Everest, all right, besides falling off the mountain or dying of frostbite or all the obvious reasons for why anybody would want to climb Mount Everest. But one of the things that occurs is as you move up in altitude for an extended period of time, the demand for oxygen for your tissues increases because the amount of oxygen in the atmosphere decreases. So your body responds by making more red blood cells. But when it makes more red blood cells, it pumps it out, and it takes some time. It's not instantaneous, okay? But over time, your blood will become thicker. And so the viscosity increases, all right? So if we want to decrease, all right, the blood viscosity, all right, we would want to decrease the formed elements or increase the fluid. All right, we'll also see, all right, the viscosity increase, like I said, in states of dehydration. All right, so let's take a look and see some of the physical characteristics of our blood, like the plasma concentration. All right, what's where, how much is present. And again, when we're dealing with the plasma concentration, we're talking about the solutes, the stuff, right? Those carrier proteins, the ions, how much dissolved items are floating around in the plasma, right? And so again, this will play a role in osmosis. All right, so the plasma, this is an important statement. The plasma concentration of solutes, this will determine the direction of osmosis across the capillary walls. This will determine the direction of where water moves. Okay, the more concentrated your blood is, remember, water is a follower. It's going to go to an area where there's more solutes. Because there's more solutes, there's usually less water. So water goes from an area of high water concentration to an area of low water concentration. So if you are dehydrated, all right, your blood or your plasma in your blood is hypertonic. If you remember that term, hypertonic, that means we have tons of solutes, all right, in that solution. And so you'll have less solutes in the tissues. So water, all right, is going to go from the tissues into the plasma. All right, I mentioned this before about temperature. It is 1 degree higher than our normal body temperature here. So that's why when blood flows restore to certain areas, okay, you'll feel a warming area there. Okay, and then of course, blood pH, slightly alkaline. This is the number range here, 7.35 to 7.45. Know that. You need to know that. And if we fall outside of that range, okay, then what we'll see is the process of denaturation, in which these plasma proteins will start to basically unfold, okay? Because a lot of times these proteins are folded in a certain configuration that helps determine their function. Well, unfortunately, if they start to unfold, their function starts to become compromised. So how do we know like what's in our blood? Simple. We're going to take a blood draw from you. I think I have a picture right here. Okay. So we're going to take a blood draw from you, and we're going to put it into a vial, and then we're going to put it into this machine here called the centrifuge, and it'll spin it for a while. Okay. About 10 minutes. And then it'll separate what we call our blood elements into the following, all right, into our plasma. Then we have a buffy coat, and then we have our red blood cells at the bottom. I'm going to come back to this other slide here. So again, that's our whole blood. We pulled it from your body, we placed it in a centrifuge, we spun it off, and we got 3 things out of it. All right, at the bottom, okay, we have our erythrocytes. That's going to be about 44% of the sample. So 44% of your blood is going to be made up of red blood cells. Yay, that's good. That's what we want. Okay. In between the bottom layer and the top layer, we have this very thin layer, okay, roughly about 1%. Call it the buffy coat, okay, because it has this appearance that's gray-white color. And this is going to be made up of white blood cells and platelets. And then on top of that, it's going to be the plasma, right? So it is going to be like a yellowish kind of color with a kind of red tint to it. So we call it straw-colored liquid. And that is going to be 55% of the sample. All right, so let's take a look at our picture here. Zoom in a little bit. All right, so let's start off here at the bottom. You can see here at the bottom, here is the erythrocytes, the red blood cells, 44% are going to be in our sample. Then you can see this small little thin layer here. There's the buffy coat. And that's going to be made up of, all right, all the different types of leukocytes, okay? T cells, B cells, all right, you're going to have the eosinophils and all that, and then our platelets. And then the remaining portion is the liquidy part. And that's going to be made up of 3 different categories. The majority of it is going to be water. So 92% of that 55% is made up of water. Okay, makes sense. And then the remaining 8% of the blood plasma is going to be proteins. And we'll go through the different types of proteins. Albumins, all right, globulins, that can include antibodies, all right, fibrinogen, that's the stuff that will help us with clotting, okay? And then, of course, we have our other solutes, the stuff that's dissolved in there. Electrolytes can be like calcium. So when you go in and you get your blood drawn and you get the results from the lab, and you'll see things on there like calcium, sodium, potassium, that is these solutes here. Those are the electrolytes. They look at glucose, all right? They'll look at lipids, all the different stuff here, all right, will be found in this area here, okay? And all of that is found in the blood plasma. So when we are spinning down the blood and we're looking at our centrifuge blood, there's a term that you'll see, and you'll see it on your lab results called hematocrit. You need to know the definition of this. This is basically the volume of all the formed elements. All right, but pretty much because the majority of those formed elements are erythrocytes. I mean, they make up 44%. And then 1% is going to be made up of the white blood cells there, okay? But that gets then broken, and platelets, but that gets broken down into the different types of white blood cells and also into the platelets there, okay? So normally when we talk about hematocrit, we're really focusing on the erythrocytes, but it does include, obviously, all the formed elements. So it will include white blood cells and the platelets, right? So normal numbers for males, 42 to 56%. Normal numbers for females, 38 to 46%. And the reason why for men ours is higher is because of our testosterone, because it stimulates, all right, erythropoietin, which I'm sure you're probably familiar with. All right, the erythropoietin is going to be that hormone that stimulates the kidney to make erythrocytes. All right, very good, very good. Okay, so now let's look at the plasma. All right, so we broke it down into those 3 components: water, plasma, proteins, and then our dissolved molecules and ions. It's an important, and I need to get this point across to you. I just don't throw stuff up here because I want to fill your brains with stuff, because this will help you later on in later chapters when we talk about fluid balance and all that. This isn't what we call an extracellular fluid. Important when you look at that term extracellular, this is part of the fluid that is found outside of the cells. We're going to learn in later chapter that your body is made up of compartments, and you can break it down into 2 basic compartments, and then we can subdivide those compartments. You have the intracellular fluid, and that's basically all the fluid that's found inside the cells. But we really can't measure that. All right, we can guess, but we can't really measure that. But we can measure the extracellular fluid. That's the fluid that's outside. Okay, so, but then when we're looking at the extracellular fluid, we have to consider what we call the interstitial fluid, and that's the fluid that's found in the tissues. You know, if you remember connective tissue, you have cells, and then you have what's called the extracellular matrix, which is made up of ground substance and protein fibers. So when we're talking about the interstitial fluid, that's that fluid that resides in that tissue there. But then also there's another compartment, and that's our circulatory system. And so that's what we're drawing the blood from, okay? And it's considered to be the extracellular fluid because it's outside the cells. Okay, so like I said, it's similar to the interstitial fluid, right? Except we're actually in the circulatory system here. So we'll see, all right, when we're talking about the plasma here, all right, our plasma will have a higher protein concentration. And that's important because proteins are going to help play a role with the movement of certain things in and out of a blood vessel, namely a capillary. Keep that in mind, all right? We'll talk more about that, but just keep that in mind. So our plasma is going to have a higher protein concentration than interstitial fluid, okay? Okay, so you've heard this term before. I hope so. If you've taken my class, you probably might remember it, hopefully. All right, blood is a colloid. And basically, a colloid is going to be something that is made up of proteins here. Okay. And that's what we're talking about when we're looking. We're going to see all these various plasma proteins that are going to be floating around in our blood. Albumin, globulins, fibrinogen, tons of different clotting enzymes, or proteins, excuse me, obviously hormones, and then enzymes that we'll need for certain physiological chemical reactions. Now, predominantly, most of these are made in the liver. So a lot of times when you get your blood results back, and if they're looking at the albumin, okay, if albumin levels are low, I mean, that's not the only thing that they do to assess the health of the liver, but if the albumin levels are low, then we start to think, okay, all right, there could be a problem with the liver. All right, so we have to keep that in mind. All right, but most of these plasma proteins will be produced, all right, by the liver. All right, so that brings me, all right, to our plasma proteins. And this is one of our big important concepts here. We're going to talk about this term called colloid osmotic pressure. All right, so let's kind of simplify that term. All right, when we're talking about osmotic, we're referring to osmosis, which means we're referring to water. In colloid, we just talked about colloid, think proteins. Okay. So the plasma proteins are going to cause this pressure to be created, right, which is going to influence the movement of water, right, from the blood into the tissues and vice versa. So the colloid osmotic pressure helps to prevent water loss or fluid loss from the blood. Think of these plasma proteins like magnets, and they're going to help attract, all right, fluid towards them. So as, remember, capillaries are very thin structures. They're one cell layer thick, squamous epithelium, okay? So for our purposes, capillaries can be what we call leaky, all right? And so you're going to send this fluid through this leaky system here, and we want to try to prevent, all right, a lot of fluid from leaking out of these structures. Well, these plasma proteins help to deal with that. They attract the fluid and help to prevent it from leaking out of those capillaries as the blood moves through those capillaries. So this is important because, one, it helps to maintain blood volume because it prevents the blood from leaking out of the capillaries, and it helps to maintain blood pressure. Okay, so this is important. So colloid osmotic pressure, it's an important term, helps to prevent fluid loss from the blood. Okay, so certain diseases can mess around with our colloid osmotic pressure. Perfect example, if we damage the liver makes a lot of these proteins. So if you have cirrhosis of the liver, all right, you can decrease the colloid osmotic pressure. So you'll see what will happen is, all right, we'll get fluid loss and we'll start to get tissue swelling. If you ever have a patient that comes in and you see bilateral, all right, swelling in their ankles, a lot of it, okay, first thought should be cardiovascular. Always, always. You should just think that because that needs immediate attention. You rule that out, then you start to think, all right, a liver disease. Okay, that's usually what you're going to be considering. All right, so for the two examples here, liver diseases, all right, will decrease the production of those plasma proteins. Kidney, same thing, all right, because unfortunately with certain kidney diseases, think of the kidneys as the filtering system of our body. They'll increase the elimination of these plasma proteins. For the most part, plasma proteins are pretty safe from elimination when they're going through the kidneys because they're big. And the filtering system in the kidneys is designed to filter out really small things. All right, but if we start to damage that filtering apparatus, then larger proteins will slip through and they'll get eliminated in your urine. And so if we have those plasma proteins getting eliminated in the urine, we have nothing to pull back on the fluid in your blood, and you'll start to leak it out of the capillaries into the tissues. All right, albumins. These guys here are the most abundant group of our blood plasma proteins here. And so they play the biggest role, all right, in determining our colloid osmotic pressure. So like I said, you hurt this group of proteins, you're really going to hurt the colloid osmotic pressure, right? But they're transport proteins. They'll carry, right, all sorts of stuff. Lipids, for example, some, not all, but some of our hormones there, and then certain ions. And you know the importance of certain ions. For example, sodium and potassium are important ions, right? Because we need those for proper neurological functioning. All right, so these albumins will act as transport proteins for these substances here. Globulins, this is the second largest one. All right, so we have alpha globulins and beta globulins. Again, these play a role because we talked about it, chapter 17, about our water, all right, soluble molecules. Well, in this case, all right, we will have to carry, all right, some hormones, all right, that are water insoluble. All right, because they can't just go ahead and dissolve right into the plasma. So we need something to carry them around. Well, here comes the alpha globulins and the beta globulins. They'll carry some of those water insoluble molecules, some of those water insoluble hormones, metals, ions, and so forth and so on. All right, they play a role. Okay. And then finally, our GABA globulins, these guys we hear tons about, the immunoglobulins, also known as our antibodies. They play a huge role in the immune function of our body. So they're floating around in our blood plasma. So if you have damage to the kidneys, you're going to lose all this stuff. This stuff will get peed right out. All right, fibrinogen, okay, think of blood clotting. Important, okay, because, right, once injury occurs to a blood vessel and we start the clotting process, all right, we need, all right, this plasma protein, okay, to create our fibrin. And it's insoluble, which means it will not dissolve, all right, when it's exposed to a fluid solution, because that would be bad, because then our clot would go away, and then anything that we were trying to prevent being lost will be lost. All right. So when you hear the term serum, what we're referring to is our blood plasma with the clotting proteins removed. And then the last category is going to be the regulatory proteins, and that's pretty much going to be enzymes and hormones. All right. This goes back to chapter 2. All right. When we talk about solutes and solution, just to review, solute is something that you add to a solution. Think of a solution as a liquid. Think of a solute as something that you put into that liquid. Like, for example, water is our solution. We add sugar to it. Sugar is the solute, and it dissolves into the water solution here. So blood is also considered to be a solution. Okay, so it's going to have both organic and inorganic molecules. Now, what defines something as organic is it means that it has carbon in the molecular formula. Okay, if it doesn't have carbon in the molecular formula, then it is referred to as inorganic. Okay, so ions are inorganic. Right. So all these other solutes will include, all right, electrolytes, nutrients, which would be lipids, which would be just regular proteins, which would be amino acids, which would glucose, right? Our respiratory gases, carbon dioxide, also oxygen. We'll even throw nitrogen in there. Okay, waste products like urea, ammonia. Okay, all of those are going to be part of the other solutes that we're going to lump into when we're looking at our blood plasma. Right. So important to know, right? This goes back to chapter 2, polar, right, or charged substances will dissolve easily. Very nice. All right. Keep in mind, though, that polar and charged substances, all right, cannot easily cross over the plasma membrane. Nonpolar molecules, okay, do need carrier proteins. But nonpolar, and if they're small enough, they don't need help getting across the plasma membrane. They can usually diffuse right through the plasma membrane of our cells. All right, so polar and charged substances, no problem. Drop them in solution, in this case into our blood, they dissolve right away and they go circulate around. Nonpolar molecules are the ones that are going to require those proteins there. All right, hemopoiesis, important. This is when we make the formed elements of our blood. This occurs in the red bone marrow, all right, of your bones, not all the bones. All right, now when you're younger, all right, a lot of the long bones will help, all right, but as you get older, then the majority of the long bones stop, all right, with their involvement in hemopoiesis, and it's usually the flat bones and the bones of usually the axial skeleton. So like the vertebrae, the skull, the cranial bones, all right, and then the epiphysis of the humerus and the proximal epiphysis of the humerus and the femur are usually going to be the places where we're going to get hemopoiesis occurring. So this occurs in the red bone marrow, all right, and so we're going to start off, all right, with the cell line called this hemocytoblast. Those are the stem cells, and this is an important term. When you see the term pluripotent, these are the cells that can turn into almost any type of cell. Okay, a pluripotent stem cell is a stem cell that you can pretty much, all right, navigate into almost any other type of cell in this formed element stem cell line. Which means, since we're talking about hemocytoblasts, right, these are going to be the cells that are eventually going to turn into some type of formed element in our blood, either a red blood cell, a platelet, or a white blood cell. All right, so we have two different cell lines. Okay, we have our myeloid line and then our lymphoid line. All right, the myeloid line is pretty much going to form the red blood cells in all of our white blood cells except the lymphocytes, also known as T cells, B cells. And they'll also make the megakaryocytes, which are going to be the platelets. All right, the lymphoid line is only going to make the lymphocytes. Easy way to remember it, okay? Just remember that the lymphoid line makes lymphocytes and the myeloid line makes all the other cells. Easy, done. You don't have to worry about trying to figure out which is which and what is what. All right, so you have these chemicals, they're called CSFs, and they're called, which stands for colony stimulating factors. And these chemicals are going to influence, all right, and also kind of guide, all right, hemopoiesis in a specific direction. All right, so we're not just making random cell lines. All right, these colony stimulating factors are going to guide the whole process to a specific type of cell line here. All right, so let's look at erythropoiesis. How do we make red blood cells? All right, so we need some ingredients, obviously. All right, well, red blood cells are going to eventually carry oxygen to oxygenate tissues and whatnot, and we need iron, Fe, okay, because that is what the oxygen is going to bind onto. We're also going to need, all right, some B vitamins, okay, vitamin B12 is largely important. And then, of course, why not add some amino acids in there? Because, yes, we need to make proteins, right? So we need the building blocks. So erythropoiesis requires some iron, all right, a couple different types of vitamin B, namely vitamin B12, and then amino acids. So we're going to start off with our stem cell. And since we are going to make erythrocytes, it has to be from the myeloid line. Okay, so we have this, what we call multi-CSF chemical being secreted, which is going to stimulate the myeloid stem cell line, all right, to move us into the erythrocyte direction. Right now, again, I'm not going to bore you with this because this is just straight up memorization. So we're going to first form the progenitor cell, which will then turn into what we call a proerythroblast. Now, understand that the younger versions of our erythrocytes are going to have a nucleus. So this one has a large nucleus. And if you have a nucleus, that means we're going to be undergoing a lot of protein production, okay, because that's where we're going to be producing our ribosomes and start to make certain organelles. So the proerythroblast will then change into the erythroblast, which becomes smaller. And this is where we start to produce our hemoglobin, right? Then it transitions into a normal blast, and then it still becomes smaller. But here's the important thing. The nucleus is no longer with us, okay? Once that happens, all right, then that cell is not per se considered to be dead. I don't want you to think that. And I know when we talked about tissues back in chapter 5 during the keratinization process of the keratinocytes, that once the organelles in the nucleus were gone, all right, because the cell filled up with keratin, the cell is considered to be dead. All right, here, all right, the erythrocyte, the adult erythrocyte, let me restate that, okay, does not have a nucleus. Okay, so once we get to that nucleus portion, or when we're without the nucleus as a normal blast, then we're moving into the reticulocyte stage, and now we've gotten rid of all the organelles, okay, except for ribosomes. If you recall, ribosomes are these proteins that make protein, okay? Well, in this case, they're going to make hemoglobin. Okay, we need lots of hemoglobin, tons and tons. All right, there's millions of hemoglobin molecules in a red blood cell. And then from there, all right, our reticulocyte matures and it becomes a full-on erythrocyte, and then no more ribosomes because we have all the hemoglobin that we need. So sometimes if you get your blood drawn, and it might say in there on the lab report that you have an increased number of reticulocytes. Those are the baby red blood cells. They're the cells, okay, they're the cells that are just before, that's not what I want. The erythrocyte becomes all grown up. So let's take a quick peek here. Here's our hemopoiesis flow chart here. We start off with our pluripotent, all right, stem cell, the hemocytoblast, and then it can differentiate into one of two lines. The lymphoid line, which makes T and B lymphocytes, right? And then the myeloid line, which turns into everything else. So we just talked about the erythropoiesis. EPO stands for erythropoietin. That's that hormone that is going to stimulate these cells, all right, to differentiate into red blood cells, all right? So here we can see, I won't go through this all, but you can see how as we move through, all right, here's our normal blast. We no longer have our nucleus here, the reticulocyte. We have ribosomes, no nucleus, and then eventually we don't have any organelles, no nucleus. Our erythrocyte then takes on that biconcave position there, or appearance. All right, again, I'm not going to expect you to know all these steps, okay? But just know that a mature red blood cell does not have any organelles or nucleus there, okay? Now, when we talk about hemopoiesis and the formation of leukocytes, we call that leukopoiesis. All right, same thing. We have 3 different types or categories here of our leukocytes. We have what's called granulocytes, monocytes, and then our lymphocytes. All right, so when we're going to talk about the first two, the granulocytes and the monocytes, because they come from the myeloid stem cell line. All right, so granulocytes include, and we call those granulocytes because there's granules inside of the cell. So we have neutrophils, basophils, and eosinophils. All those cells end with phil. All right, so these are in the granulocyte family. Similar type of situation. All right, colony stimulating factors will be produced, and it will cause, all right, these cells to advance from the myeloid stem cell line to form the progenitor cells for whatever specific type of granulocyte that we're going to be dealing with here. And you can see right here in our drawing here, all right, when we talk about here, all right, our granulocyte cell line, then we have our monocyte cell line. So our granulocyte cell line here, all right, you could see, again, just different colony stimulating factors, all right, will be produced that will guide, all right, the formation of our white blood cell into a specific type of granulocyte, be it an eosinophil, a basophil, or a neutrophil. Then meanwhile, okay, we'll have a specific type of colony stimulating factor, which will then guide our progenitor cell towards the monocyte line. Then our monoblast will turn into a monocyte eventually, but this eventually transforms into a macrophage. A monocyte is a macrophage in circulation. Okay, it hasn't turned into the macrophage yet. It's just circulating around, all right? And then it'll eventually become a macrophage when it arrives at its destination somewhere in the tissue. Okay, so when we're talking about our granulocytes, the progenitor cell will first become a myeloblast, and then it'll form one of the three types of granulocytes, a neutrophil, basophil, or eosinophil. And then, all right, well, if we're talking about the monocyte line, okay, again, we're still dealing with the myeloid cell line. All right, this, real simple, this type of stem cell, once we have the monocyte colony stimulating factor being released, that will then tell the progenitor cell to become a monoblast. That monoblast will eventually become a monocyte, and that monocyte will eventually become a a macrophage. So coming back here, this is what we're talking about. So basically, we have our progenitor cell, and depending on which of our colony stimulating factors is produced, it's the granulocyte one or the monocyte one, will determine which direction the progenitor cell goes in. Okay, and then that will eventually form one of three basophils if it decides to go to the granulocyte line. Right? If it goes towards the monocyte line, then it's eventually going to become a monocyte, which will eventually become our, there we go. It'll eventually become our macrophage. All right, leukopoiesis. Remember, leukopoiesis, all right, has to do with the leukocytes. And part of our leukocytes are our lymphocytes. But our lymphocytes come from that other type of stem cell, all right, called the lymphoid stem cell line. All right, real easy. This one's basic because you have two types of lymphocytes. You got a B lymphocyte and a T lymphocyte. So same type of scenario, okay? The lymphoid stem cell, okay, will either differentiate into what we call a B lymphoblast or a T lymphoblast. If it differentiates into a B lymphoblast, it will eventually become a B lymphocyte. If it differentiates into a T lymphoblast, it will eventually become a T lymphocyte. All right, and what's really cool is some of these cells, these lymphoid stem cells, will go right to what we call natural killer cells. And that's just a cool name in itself, right? But that's what they do. These natural killer cells, and we'll talk more about them in our, when we get into the immune system here, okay? So here you can see we have our lymphoid cell line, okay? And so that those cells can either become a B lymphoblast or a T lymphoblast, or skip jail or whatever and go right to natural killer cell. But all of these are lymphocytes here. B lymphocyte, T lymphocyte, and then natural killer cells. You may have heard of T cells and B cells. That's what they're referring to when they're talking about those cells. T cells and B cells are the lymphocytes here. All right, but again, we'll talk about the functions of those cells when we get to the immune system. And then, of course, we can't forget about our platelets. And then we have our thrombopoiesis. How do we make the platelets? We're going to start off with this big, huge, massive cell called a megakaryoblast. Again, it comes from the myeloid stem cell line. Okay. And so, again, we'll have this huge cell, and we'll have a chemical exerted on it called thrombopoietin. And what this does, and it's kind of neat how this does this, but what happens is this megakaryocyte, okay, is basically going to get really close to the cell wall, not the cell wall, the vascular or blood vessel wall, all right, and it will start to push parts of itself, all right, called proplatelets, which are just these long extensions of the cell. It'll start to push these long extensions of the cell into the bloodstream. And as the blood is flowing by, it will slice off fragments from this huge cell. And those fragments become the platelets. If you ever look at a blood smear, platelets are really small. Well, let me go back to this one. Hold on. Just real quick here, you can see, again, we won't go through all the fun stuff. Here's that thrombopoiesis, right? And it turns into this megakaryoblast. Then eventually it differentiates into a couple different types, but then eventually it turns into what we call a megakaryocyte. And the megakaryocyte, all right, will have these extensions here. I'll show you another picture in a second that, all right, these proplatelets, these extensions squeeze in between some of the cells of the blood vessel wall, and the blood, as it's going by, just rips off or slices off parts of these proplatelets, and they turn into platelets. That's what this picture here on this slide is showing you. Okay, see, you have these nice proplatelets just squeezing through, and as blood is flowing through, it just slices off these pieces, and they turn into the platelets there. That's basically how that one works. I know that this is not the most exciting stuff to talk about, but again, you know, we do have to kind of talk a little bit about it. All right, so it helps you with your understanding a little bit more. All right, erythrocytes, red blood cells. Let's get into talking about them because it is important. A lot of the physiology, especially when we get into respiratory physiology, is important that you understand about what an erythrocyte looks like and what's included in an erythrocyte. Okay, so first and foremost, you have to understand that erythrocytes have to be really small and flexible, okay, because they got to squeeze through things. So that's very important. In fact, erythrocytes have calcium ion pumps in their plasma membrane because calcium has this property in which if calcium starts to accumulate in certain things, structures, areas, what have you, it starts to become hard. Look at your bone, for example. When we deposit calcium in the osteoid, all right, the osteoid starts to stiffen and become hard. Calcium deposits in plasma membranes of red blood cells, those plasma membranes can become stiff. And if they become stiff, when these red blood cells have to squeeze in and out of areas, okay, then they become damaged and then they become useless. So they're small and flexible. We already talked about how they do not have a nucleus and they do not have organelles, okay? The reticulocytes have the ribosomes, okay, but they don't have a nucleus. Right, but the mature red blood cell has none of that. Okay, the only thing it has is this hemoglobin. That is the that is the oxygen carrying molecule of a red blood cell. So when we look at a red blood cell, it looks to me like a donut from the side. We call it a biconcave disc. So the center is sunken in. So when we're looking at it from the side, it looks like that. When we look at it from the top, the area here is thinner, appears lighter because it's thin there, okay? All right, so it is because of this appearance here, all right, that our red blood cell is able to move in and out of things. You know, it has this shape, all right? We have that spectrum protein there that helps to make it flexible. We have our calcium ion pumps that keep calcium out of the red blood cell so it doesn't stiffen up on us. And because of these structures here, we're going to see, especially in some of the really small blood vessels, the capillary, which is the smallest of our blood vessels, All right. At one point in the capillaries, there's just enough room for one single red blood cell to make it through. Well, what will happen is we'll see these red blood cells start to stack up in a single file line. You remember when you were in elementary school and they made you walk everywhere in the building in a line? They always said single file, single file. All right, that's what we see here with the erythrocytes. We have the single file line, okay, we call that a ROLU. And that's because it is, I'll show you, such a small, here you can see it. Here's our capillary right here. And you have all these red blood cells just stacked up on one another in that ROLU formation there. So as they're moving through. And blood flow starts to slow down a little bit. We'll talk more about that later on. All right. We want it to slow down a little bit so oxygen can leave and go into these tissues, and then carbon dioxide can exit the tissues and enter into your capillary there. All right. So we want that to happen. Okay. So it's important that when we talk about the function of red blood cells, right, this is going to involve the transportation of our respiratory gases, oxygen and carbon dioxide. It's just a transport vehicle from point A to point B. It's going to take oxygen from the lungs to the tissues. It's going to take carbon dioxide from the tissues back to the lungs. That's it. Don't overthink that. All right, so you can see, now I said over 1 million molecules, all right, I well understated that. 280 million hemoglobin molecules are in, all right, our erythrocyte. That's a lot. That is a lot. Now, I want to blow your mind even more. 280 million hemoglobin molecules, and one hemoglobin molecule, okay, can hold on to 4 oxygen molecules. So do the math. I'm too lazy to do it. I did it one time and I can't remember the number. But one single erythrocyte can carry, what's 28 times 4? 56. Yeah, it's over 1 billion, I believe. If my math is correct, that's over 1 billion oxygen molecules. That's a lot. All right, so what is hemoglobin? Again, it is the transport vehicle inside the red blood cell that is going to carry oxygen and carbon dioxide. Okay, when our hemoglobin molecule, remember what I said? You can have 4 oxygens on a hemoglobin molecule. Okay. When the hemoglobin molecule is pretty much loaded up with oxygen, we call it oxygenated. Okay. When it has very little to none, all right, then it's deoxygenated. So our hemoglobin molecule is a protein that gets folded up. Okay. It's made up of 2 alpha chains and 2 beta chains. Again, not terribly important for you to memorize that, just be familiar, but they're globulin, protein molecules. And so they're folded up, okay? And at the center, all right, of these chains here, all right, we have a heme group. And what's important about that is that it has an iron ion molecule at the center of that group there. And that is what binds the oxygen, okay? You need to know that. The oxygen binds onto the iron right here. That's important. Star that. So like I said, each hemoglobin can carry 4 oxygen molecules because you have 2 alpha chains and 2 beta chains. And each of those chains has an iron ion in the center, and that's what oxygen binds to. Pretty cool. Pretty cool, pretty cool. I like that stuff. All right, hemoglobin. Know this. Hemoglobin binds to iron directly, okay? It is a weak binding, which is good. Believe it or not, this is the first time you probably ever heard somebody say that weak is good, but it is good because we don't want it to be a strong binding because then we have to fight to get the oxygen loose. And all this stuff happens so fast, okay, because blood flow is always in motion, okay? It's always in motion. So we need rapid attachment. So what we'll see is if the binding is weak, then it can come off easily. And we need that to happen because when we go to the lungs, we want to pick up oxygen and get rid of the carbon dioxide. So we want, and when we go to the tissues, okay, we want to offload that oxygen. So if it's a strong bond, we have to fight with it. If it's weak, comes right off, goes into the tissues. Bye, see you later. Okay, now, important to know, carbon dioxide does not, I repeat, it does not bind to the iron. It's going to bind onto the globulin protein itself. Again, weak, which is good, because everything's happening fast. Rapid attachment, right? So we wanted to detach that carbon dioxide from hemoglobin when we get into the lungs, so we can offload it, so you can exhale it, get rid of it. Only time, or one of the few times you'll ever hear me say that binding or weak binding is good. So here you can see, all right, our hemoglobin molecule here. All right, so what we're looking at here at the center of our chain here, that's, I'll zoom in, that's iron. And that's what oxygen is going to directly bind onto. So if you want to look at the whole molecule itself, our hemoglobin molecule, you can see it's made up of those 4 chains. And what we've done is we've just kind of folded them together in this proper configuration. Now, mind you, to kind of give you an idea, all right, if we started to make our blood acidic, okay, our pH, say, drops down to like 6.8. Might not seem like a lot, but it's not good, right? And what will happen is these alpha and beta chains will start to unfold, and it starts to change the configuration of that molecule, and it basically will start to make it useless, and then we can't bind oxygen, and that's a problem, okay? So keep in mind, the iron is where the oxygen binds to, the carbon dioxide is going to bind right onto the chain itself. Which brings me to, and you guys remember this from chapter 17, erythropoietin, also known as EPO. This is going to stimulate, all right, the process of erythropoiesis. So as you recall, all right, the kidneys will predominantly produce the erythropoietin. And what stimulates, all right, let's go back to our homeostatic mechanism, all right, that reflex there, the stimulus is going to be decreased blood oxygen levels. Okay, so the blood oxygen levels go down, and so the red bone marrow starts to kick into, I won't say overdrive, but it turns on, the machinery turns on in your red bone marrow, and it stimulates the myeloid cell line, okay, to start to produce erythrocytes. All right, so EPO, erythropoietin, stimulates the red bone marrow to produce the myeloid stem cell line. And it makes erythrocytes, which will then be deposited into your circulation. And as we increase the number of erythrocytes, we will then increase the oxygen carrying capacity of your blood. Now, this is based on a negative feedback system. So once it gets to the acceptable level, when it gets high enough, all right, then we'll shut down EPO. The kidneys will stop producing erythropoietin, and then that will shut down the red bone marrow. All right. Now, we'll also see testosterone. Okay, like I said earlier on in today's lesson, that testosterone will help to also stimulate EPO. And a lot of that has to do with growth. All right, and then what I had to write a paper on, I actually wrote a paper on erythropoietin when I was in med school. So I learned quite a bit about it. Fascinating stuff. But I used that example before about climbing Mount Everest, and certain factors, okay, such as altitude, will also stimulate, all right, erythropoietin levels. Some athletes will train at higher elevations, I won't say right before a competition, but close to a competition, because it will stimulate the production of erythropoietin. And if it stimulates that erythropoietin, they'll increase their oxygen, their blood oxygen carrying capacity right before a competition. So they'll have more oxygen on hand. It's almost like a natural form of blood doping. Problem is, you got to be careful, because if you make the blood too viscous, too thick, you can get clotting and damage to blood vessels, and we don't like that. So you've seen this picture before, all right, about erythropoietin production. Here's the stimulus. We see decreased, all right, blood oxygen levels. The kidneys then release the erythropoietin into the blood. The erythropoietin then stimulates the red bone marrow, all right, to produce erythrocytes, and then the erythrocytes get dumped off into the blood, therefore increasing, all right, the blood oxygen carrying capacity. And then by the negative feedback system, that will shut off the kidneys from producing more erythropoietin once you get to the acceptable levels. And that's the beauty of the negative feedback loop. All right, so like I said before, erythrocytes only live for about 120 days, okay? And so because they only live for 120 days, all right, we have to, keep renewing them, all right? And part of the reason why they don't live for 120 days is they have no nucleus. And if you don't have a nucleus, you're not gonna be able to produce ribosomes to help make proteins. And if you don't have proteins, we can't repair the cell, all right? And they don't have any organelles. So where do they go to die? Okay, they go to the liver or the spleen. And so they'll go to one of those two organs and become phagocytized. And it's kind of cool how your body does this, right? Because they'll break down, all right, the red blood cells, and then they like to recycle things, as do a lot of the processes in your body do. They like to recycle things. So they break down these red blood cells through phagocytation. All right, and we'll see, all right, amino acids coming from these guys. And so we can use those amino acids for more protein synthesis, maybe to make more red blood cells or for something else. But there's other components to the red blood cell. Remember the hemoglobin molecule. We had iron from the hemoglobin molecule, right? So we have a transport protein in your blood called transferrin, okay? And so that transferrin is going to pick up that iron and it's going to ship it over to the liver. So we can use it for an abundance of other processes. So if we don't have anything that we need it for on hand, no problem. We'll store it, okay, with some of these storage proteins, hemosiderin and ferritin, all right, for another day to make something else, okay? Maybe we just don't need any more red blood cells at this time, but when the time comes, all right, these storage proteins then, all right, can then migrate over to the red bone marrow so we can make more erythrocytes. So it's really kind of cool how when our erythrocytes are broken down, how we can make more. So this is interesting because this is where we're going to get into some terms here that you may have heard before and you just never knew what was the clinical consideration for this. All right, so when the erythrocytes are being broken down, okay, we're going to deal with the hemoglobin molecule. All right, we've already taken care of the iron, so we're going to just snap that off, bind it to ferritin and ship it off to the liver to be stored. Okay, but what about the rest of it, the rest of the heme? Okay, well, we have our lovely macrophages that once used to be monocytes. And so they'll start, like I said, through phagocytosis, they're going to break down the heme group, all right, into a substance called biliverdin. Now that sounds familiar. That probably sounds familiar to this term here, bilirubin. Yes, you are absolutely right. Bilirubin is an intermediate molecule that comes from the destruction of red blood cells, namely the heme group of red blood cells. We're familiar with bilirubin's yellowish pigment because you might see it in somebody's skin. We see it in newborn babies sometimes, okay? But biliverdin gets converted into bilirubin, and bilirubin gets taken to the liver through one of our protein, plasma proteins, albumin. And so it gets taken to the liver, and we're going to deposit it into bile. Remember bile? Okay, some of you are familiar with the gallbladder. Okay, you may have lost your gallbladder. But see, it's the liver that produces the bile, and it's the gallbladder that stores and concentrates it. So we're going to use this bilirubin, all right, and make it part of the bile, and we'll dump it into our digestive system. Now, when the bilirubin makes it to the small intestine, it gets converted into a different substance called urobilinogen. And it's this urobilinogen that, again, is going to work its way through our digestive system, through the small intestines into the large intestines, and then it gets converted one more time. But we need the normal bacterial flora there to help convert it, all right, into what we call stercobilin, which gives our feces that brown pigment. A lot of stuff goes on to that heme group, all right? So that heme group gets converted to biliverdin, which has a greenish pigment. Then it gets converted to bilirubin, which will then be incorporated into our bile in the liver. And then when that bile is released, all right, into the digestive system, okay, that bilirubin gets converted into that urobilinogen. And then eventually it becomes that stercobilin, which gives us that brown pigment there. And then what gives us the yellow pigment to our urine is when, all right, the urobilin gets absorbed back into the blood, and then it gets converted into urobilin, gets filtered out or excreted by the kidneys, and that's what gives us our yellowish pigment there in our pee, in our urine there. So it goes through a lot of stuff happens there. All right, so let's talk a little bit about blood types, and I'll finish up with that for today. And some of you are probably chomping at the bit like, yes, blood typing. I want to know about blood typing. Okay, well, let's talk about blood typing. So when we talk about blood types, and I keep forgetting what I am, I'm almost positive that I'm A positive. So let's just say I am A positive. When we talk about blood typing, what we're really talking about is our surface antigens, okay? So what is an antigen? An antigen is this. It's a glycoprotein. It's a protein with a carbohydrate group attached to it, all right? So when we talk about blood types, what we're talking about is these surface antigens, these glycoproteins that sit on the surface of our red blood cell, on the plasma membrane. So we call it ABO blood typing or blood grouping. And so when we're discussing this, what we're referring to is, right, we're going to either talk about the presence or the absence of one of these antigens. Is it the A antigen? Is it the B antigen? Or is it both? Or is it the absence of those? Okay, so let's talk about this. If you are A positive, which I am, okay, to the best of my knowledge, all right, that means that on all my red blood cells, I have the surface antigen A only. Okay, so your blood type tells you what surface antigens you have. So obviously, if you're B, then you have the B antigen. If you are AB, you have both antigens. You have the A antigen and the B antigen. If you are type O, think of it like 0. You have nothing. You have no surface antigens. It's important that you understand that because you're going to get confused here if you don't understand that concept. Okay? Type A antigen only. Type B, B antigen only. If you are AB, you have both antigens. If you are type O, I think of it as 0, you have nothing. Neither antigen is going to be present. Okay, so when we talk about our blood grouping, if we're referring to the person's antigen status, all right, this is going to help us when we're trying to understand their antibody status. So these first 2 bullet points are crucial for you to understand. Okay, so a person's antigen status determines their antibody status. So anti-A antibodies will react with the surface antigen A. Anti-B antibodies will react with the surface antigen B. So a person, this is important, a person, all right, doesn't have antibodies for their own surface proteins. Well, that makes sense, right? Why would you have antibodies that would attack your own cells? Since I am A positive, don't worry about the positive part. Since I am A, type A, okay, that means I have the antigen, the surface antigen A. So I will not have anti-A antibodies. Because if I did, then my own antibodies would be going after my own cells. Okay? But here's the important thing. A person does have antibodies to the antigens that are foreign to them. So because I am blood type A, I have anti-B antibodies. Okay, so here's me, type A. I have anti-B antibodies. Now, if I am type A, B, same goes for type B. I'm assuming that you can figure that out. Type B blood has anti-A antibodies. Type AB blood, remember, this has both the surface antibodies, or excuse me, antigens. So that means that this person will have neither anti-A or anti-B antibodies. They have neither. And then finally, type O is going to have, remember, type O doesn't have any surface antigens on their red blood cells. So that means that they will have both anti-A and anti-B antibodies. Okay? It's important that you understand that. This can be confusing. I want you to keep it simple. Simple, simple, simple. Okay? So let's talk about this. I like this little chart here. So here, we'll use me as the example. Here's me. I'm type A. And so I have surface antigen A. And that's represented by those little balls there. My blood plasma contains antibodies that are called anti-B antibodies. So if they come in contact with a surface antigen that has this wedge shape on it, they'll attack it. Well, that's only going to be found in blood type B and blood type AB. So I can't get blood transfusions from either one of those people. I can't, because my antibodies will go after those antigens there. But I can get type O. That's why everybody loves type O. Because look, they don't have any surface antigens. None. Okay. All right. So when we talk about the positive now, remember, I'm A positive. When we're talking about the positive, we're talking about what we call Rh factor, also known as surface antigen D. Now we're just adding another antigen to our blood typing here, okay? So this is what this Rh factor is going to determine if we are blood type positive or blood type negative, okay? Keep that in mind. So normally, all right, the antibodies, all right, are not going to be there. That's an important concept. Antibodies to Rh factor, not usually there. Okay. They get there when an Rh negative person is exposed to Rh positive blood. And so we see that with pregnancy quite often. Okay, so mom, okay, she could be Rh negative, okay, but the fetus could be Rh positive. Now that's okay for that fetus initially because mom doesn't have the antibodies yet, okay? So once the baby is born though, okay, mom now has, okay, antibodies now. So she has to be careful the second time she gets pregnant because if that fetus is Rh positive, her antibodies could attack the fetus's red blood cells. All right, so that's why, all right, they'll do the blood typing and they'll give mom, it's called Rogam. And it's this medicine that helps to prevent mom's antibodies from going after the fetus's Rh positive blood. Okay, so that's why we usually, when people are talking about blood typing, we always will report, all right, all of that together. Either they're AB, AB, or O, and they're either positive or negative. All right, so what happens if we give somebody the wrong type of blood? All right, say I got type B or AB blood. That could be a problem for me because now we're going to see I am going to start to have the process of agglutination. And this is basically where my antibodies are going to go after the red blood cells that were transfused into my body, and we start to see clumping. That's not good because we can get blood clots and blocked blood vessels that can cause tons of issues, as you know, but we can also get hemolysis, and you know what lysis means, to break down, okay? These red blood cells can just start to splice and break apart, and it can shred through the tissues of certain organs, and we can get organ damage. Here's a nice picture, okay? So type B person gets type A blood. Type A blood, all right, has the surface antigen of A. Type B person carries anti-A antibodies in the plasma. And those antibodies will glom on, all right, to those red blood cells, and it causes this clumping and clotting going on. Right? And it causes that hemolysis, and that causes some issues. So how do I know? To test, all right, before I give a transfusion, I take a little bit of blood, I put it on a microscope slide, and then I'll take the other person's blood that I'm going to give, transfuse the blood to, and I mix them. And if it looks like this, okay, no clumping, so it's negative. That means we can give you the blood. If I see this, the clumping and the lysis, that's not good. And that's what we're seeing here, that agglutination there. We don't want to mess with that. All right, folks, I'd love to keep going, but I know that some of you are probably like, please, Dr. Koss, please stop. And I will stop. But what I would like to do is I will do my best to